Hello everyone. Today we will discuss about eukaryotic cell. This is a model of eukaryotic cell found in human, animal, plants and fungi. Eukaryotic cell differ from prokaryotic cell in that they contain nucleus and other membrane bound structure called organelles. Eukaryotic cell can be plant, animal or fungi. Many single celled organisms like protozoa and all multicellular organisms are eukaryotes. Now we will discuss about the organelles of eukaryotic cell one by one. Next one is centrioles. Here we can see the structure of centrioles. Centrioles play a crucial role in cell division, mitosis, and the location of the nucleus. The centrioles are a pair of structures on either side of the nucleus made up of 9 triplets of microtubules. Centrioles play a key role in the formation of the mitotic spindle during cell division as well as helping determine the spatial arrangement and location of the nucleus within the cell. Centrioles are found in most animal cells but are absent in plants and fungi. The next structure is rough endoplasmic reticulum. The rough endoplasmic reticulum manufactures and packages protein. The endoplasmic reticulum is a contiguous complex network of interconnected tubes and membrane sacs that extend throughout the cell cytoplasm. This complex makes up roughly one half of the membrane tissue of the cell. There are two types of endoplasmic reticulum, the rough and the smooth. The rough endoplasmic reticulum or the rough ER gets its name from the fact that its surface are studded with ribosomes and the membrane of the rough ER is contiguous with the nuclear membrane as well as that of the smooth endoplasmic reticulum. The rough endoplasmic reticulum primarily function are to manufacture and then fold proteins into their final useful configuration. The proteins it produces are then either secreted from the cell, transported to location within the cell where they are needed or sent on to the Golgi apparatus for further processing. The next structure is lysosomes. Lysosomes act as the digestive and waste disposal system of the cell. The lysosomes are tiny sac-like structure filled with enzymical hydrolysis like vesicle. Lysosomes are constructed of a phospholipid bilayer similar to the cell membrane. The lysosomes function as the digestive and waste disposal organelle of the cell and enable the cell to utilize nutrients. Lysosomes also break down foreign substances such as invading microbes, thus freeing up their base component for reuse. Lysosome may act as a bandage and help to repair damage to the cell membrane caused by invading microbes. When the cell dies, the lysosomes will destroy the cell through a process called apoptosis. The next structure is vesicle. On the screen, we see the structure of vesicle. Vesicles store and transport substance within the cell. Now, we will discuss about the nuclear membrane. The nuclear membrane separates the nucleus from the rest of the cell. The nuclear membrane consists of two lipid bilayers, the inner nuclear membrane and the outer nuclear membrane. The function of the nuclear membrane is to separate the nucleus and nucleolus from the rest of the cell contents. The nuclear membrane is embedded with specialized protein called nuclear pores which act as specific doorway that permits substances such as nutrients, waste and cellular information to pass into and out of the nucleus. The next structure is mitochondria, powerhouse of the cell. The mitochondria act as a power plant of the cell. Mitochondria are the power plant of the cell. They convert sugar into adenosine triphosphate ATP, an enzyme related to cell metabolism. Mitochondria are unusual in that they are surrounded by two membranes and contain their own independent genome. They also can divide independently of the cell in which they reside, meaning that mitochondrial replication is not tied to the cell division. The number of mitochondria found in a cell varies by the type of cell. The more energy a cell needs and the more mitochondria it will have. Many cells contain only the mitochondria, while others may contain as many as several thousand mitochondria found in both plant and animal cells. The next structure is nucleolus. The nucleolus' main function is to form ribosome. Nucleolus is a dense spherical non-membrane bound structure found in the nucleus of the cell. It is composed of primarily of protein and nucleic acid. Its main function is to transcribe ribosomal RNA and combine it with protein to form ribosome. The nucleolus also can capture and immobilize protein, thereby preventing cellular interaction with their building partners. This helps regulate cellular activity. The next structure is ribosome. Ribosome reads instructions from messenger RNA to synthesize protein. The ribosomes are the large complex molecular machines found within all living cells. Their primary function is to synthesize new protein by reading the instructions from a messenger ribonucleic acid RNA strand. Ribosomes do this by clicking together amino acid in the specific sequence. Ribosomes consist of two subunits, each made up of RNA and protein. The smaller subunit reads the instruction from the messenger RNA and the larger subunit assembles the amino acid into the chain. When the ribosome completes the assembly of the new molecule, the two subunits of the ribosome splits apart. Ribosomes are the only organelles that is present in all cells, including bacteria. Our next structure is chromosome, the bundle form of the DNA strand. Inside each human cell, there is approximately 6 feet of DNA existing as 46 chromatin strands. Normally, this DNA disappears in somewhat loose configuration. However, when the cell is preparing to divide, each strand of DNA is condensed by a factor of 10,000 times into a structure called chromosome. This folding and repackaging serves to facilitate the process of the cell division. When the cell divides, the chromosome separate, providing a complete copy of the genetic information to each cell. Next one is cell membrane. Encloses and protect the cell content of the cell. The cell membrane physically separates the interior of all cells from the outside environment. It is made up from a phospholipid bilayer embedded with specialized protein. This specialized protein acts as a doorway into and out of the cell. The cell membrane is selectively permeable to ion and organic molecule and controls the transport of those substances into and out of the cell, and is involved in a variety of cellular processes such as cell adhesion, ion conductivity, and cell signaling. It also serves as the attachment surface for both internal and external structures like the cell wall and cytoskeleton. The cell membrane is critical for maintaining the internal balance of the cell. The next one is nucleus. Nucleus contains the cell's genetic material. The nucleus is a membrane-bound organelle that contains most of the cell's genetic material. The interior of the nucleus does not contain any membrane-bound organelles or compartments. Its contents are non-uniform and consist of a number of subnuclear bodies like the nucleolus. The genetic material in the nucleus is organized as multiple linear DNA molecules combined with a variety of protein to form chromosomes. The genes within this chromosome are the cell nuclear genome. The function of the nucleus is to maintain the integrity of these genes and to control the activity of the cell by regulating the gene expression. The nucleus can therefore be considered the control center of the cell. Now we can see the cytoplasm. The cytoplasm is a semi-fluid gelatin-like structure that found the organelles inside the cell. The cytoplasm is a semi-fluid gelatin-like substance that surrounds the organelles inside the cell. It acts to cushions, protect and supports the organelles and fills all the empty space within the cell membrane not occupied by organelles or other structures. Although cytoplasm is made up of mostly water, its structure and properties are not fully understood. There are multiple levels of organization within the cytoplasm, including concentration gradient of ion and complexes of metabolic enzymes. The concentration of ions such as sodium and potassium are different in the cytoplasm than in the extracellular fluid. The difference in ion levels are important in processes such as osmoregulation and cell signaling. Now we can see that smooth endoplasmic reticulum. The smooth endoplasmic reticulum synthesizes lipid, phospholipid and steroid. The endoplasmic reticulum is a contiguous complex network of interconnected tubes and membrane sacs that extend throughout the cell cytoplasm. This complex made up roughly one half of the membrane tissue of the cell. There are two types of endoplasmic reticulum, the rough and the smooth. Now here we can see the structure of cytoskeleton. The cytoskeleton gives support and structure to the cell. It is a structure of Golgi apparatus. Golgi apparatus modified protein and sent to the other part of the cell. The Golgi apparatus consists of stacks of folded membrane called cisternae. Each disc-like cisternae contain different enzymes which selectively modified the micromolecules that are passing through them. The Golgi apparatus' main function are to store, modify, sort and package micromolecules synthesized by the rough endoplasmic reticulum and ship those molecules by vesicle to destination both within and outside of the cell. The Golgi apparatus could be considered the post office of the cell. It is a structure of nuclear pore. Nuclear pore act as doorway between the nucleus and the cytoplasm. Nuclear pores are hollow protein structure that penetrate the nuclear membrane and act as doorway between the nucleus and cytoplasm. They allow small molecule and ions to freely pass into or out of the cell of the nucleus, but also may allow larger protein to enter the nucleus from the cytoplasm if the protein has special sequences or tags that indicate they belong in the nucleus, in the same way as the protein are allowed to enter RNA transcribed in the nucleus, and protein destined to enter the cytoplasm have export sequences that tag them for release through the nuclear pore. This is a eukaryotic cell. We discussed the each and every organelle of eukaryotic cell. This will prove helpful to you and boost your preparation. If you like this video, then like, subscribe and share. And comment for new 3D animation video.